here, this is the embryo, this is the young sporophyte, or young sporophytic plant. The sporophyte is just this thing, that little round, oval-shaped, ball-like structure that you see right there. And you're going to see this in today in lab. When we talk about the bryophytes, a sporophyte has three parts to it. So the sporophyte, which is two ends, it's got three parts to it. It's got the part that connects it to the female plant. That's where it's going to get its nourishment and its hormones. That's called the foot. So you see that thing at the top, the foot. So there's three parts to a typical moss sporophyte that you want to look for in all of these things today, actually. So the foot is where nutrients and hormones are exchanged. And there's I'll put it here in quotes because it's not a real placenta. Placenta is a tissue nourishment for humans. So I'll put that in quotes. It's not really a real placenta. But what happens is the foot from the sporophyte pushes itself into the tissue of the female plant, and there's a region of cells which is called the placenta, where the nutrient exchange takes place. So if you want to grow and you're non-photosynthetic, the sporophyte in this case is non-photosynthetic. It needs to get nutrients from the plant from its photosynthesis, and it needs hormones to promote cell division, which is growth and development. The next thing is a little stalk called the seta. And the stalk or the seta is this one. It's very short here in this sporophyte. So it's really just a little stalk. It's larger in the life cycle we're going to do that you'll see. And then the final part is the sporangium. R. The sporangium is also sometimes called a capsule. Like a pill, a capsule or a pill that you take for medicine, because a lot of the sporangia on the mosses looks like a little pill or a little bit of a capsule. So what the sporangium does is make spores, and this is where meiosis takes place. So meiosis takes place in the sporangium where the spores are out there. And so that's what we've got here. We've got a mature sporophyte that's about 1 millimeter in size, and it's very short-lived. It gets its nutrition from mom, the female plant. It gets its hormones from there, and that's where meiosis takes place. And those spores get released once the wall of the sporangium cracks or splits open. So once that wall splits open, they call that to dehiss. So once the wall splits open, those spores go flying out from there. So this is a heteromorphic, heterophalic sporic alternation of generations, because the two generations look different. Here's the gametophyte generations. They look different than this little 1 millimeter size round ball from the sporophyte. Heterophalic is because the two means dioecious. Heterophalic means dioecious, which means two houses or different phalli. And we have the males and females on different phalli, different houses. So that's heterophalic. Heteromorphic because the two look different. And then it's a sporic alternation of generations because spores are made as a result of, you see here, of meiosis. So that's kind of a summary of it. You're not going to have to draw this one on the test. This is just up here because you're going to be looking for all these structures. You're going to see the antheridial head, archegonial head, archegonia, antheridia. You're going to see those spores. You're going to see the sporophyte in the slides that we have for you today. This is a close-up. This is a magnified view of this right here. And you can kind of see it in there, but see these little coiled up structures? These are called elaters. And in your description of the group, our These elaters are little coiled up structures, and they're called hygroscopic. So what happens is when the sporangium, that tiny little 1 millimeter little ball, that little sporophyte, when the sporangium dehisces or splits open, air comes into the sporangium. And when air comes into this sealed structure, it causes these elaters to dry out. So these elaters are very sensitive to atmospheric moisture. So any water, atmospheric H2O that's present there, very sensitive to that. So when the sporangium dehisces or splits open, and the air comes in, it causes these things to dry out. Right now they're very long, but once they start to dry out, they coil up, they shrink. When things dry out, they kind of shrink and coil. And because they're twisted, they shrink and coil, and they fling the spores, which are in red and green here. They'll fling those spores out from the sporangium. So really their function is dispersal. You'll look for them today. So on to the second group. So that was Marchantiophyta, just a brief summary of their life cycle. The second group is, the name actually means moss plants. Bryophyta is the phylum name. And these are a little more complex in that the sporophytes do have stomata, which can exchange gases. If you remember the last group, they just have, pores, so there's a little opening where gases come in and out of there. In addition, this group does not have it doesn't have vascular tissue, but this group has conducting tissue. And the conducting tissue is called leptoids and hydroids. So lepto means slender, hydro means water. These conduct kind of what xylem and phloem conduct in a plant. Leptoids conduct mostly carbohydrates. So they'll conduct carbohydrates like phloem in plants. It's not phloem, but it conducts and behaves like phloem. Hydroids conduct, as the name implies, water and minerals. So if you conduct water and minerals, then you're behaving like the xylem, and that's what the xylem is real vascular tissue. So these are not true xylem and phloem, or phloem and xylem as I've written them here, but they do conduct carbohydrates, they do conduct water, which functionally they're the same thing, but structurally they're different. And so we'll talk about vascular tissue really next week. The gametophytes are leafy. So here they are. Here's some gametophytes with these leaf-like structures. These are not leaves. So these are not leaves. They don't make roots. If you make real leaves, that means you need to have vascular tissue running through those leaves, and you need to have certain type of structure, like we talked about when we did photosynthesis. We talked about the epidermis, mesophyll cells. We talked about xylem and phloem, and what that all means, and the vascular tissue, the xylem and phloem going into that. They don't have roots either. So to be a real root, you need to have root hairs for water absorption. You need to have xylem and phloem going into your roots, so you can then conduct in and out of there, the water and the carbohydrates. So going back to here, they don't have real leaves, they don't have real roots. They have these leaf-like structures, and they're called, they don't call them leaves again, they're just called phyllids. So a phylid is a leaf-like structure. For photosynthesis in the bryophyta. So it's for leaf-like structure in bryophyta. Its function is photosynthesis. So the tendency is for people to call them leaves, but like I said, they're not structurally. Functionally, they're leaves, but they're not structurally leaves because they don't have all the typical characteristics of a true leaf. So this is what you see there. Rhizoids are multicellular. This is different than the last group. The last group had these rhizoids. Rhizoids are for attachment, and the rhizoids function to hold the plant in the soil. These are multicellular rather than unicellular. Sporangia with dehiscent capsules. So dehiscent means to split open at maturity. So that means they split open, they crack, they open up at maturity. And so the capsules you'll see in lab, you can look at a section of those as well in this group. It looks like a little pill. You'll see it. Most cells with numerous chloroplasts, which is just like the last group, Argentiophyta. So they're photosynthetic cells in those fillets contain many chloroplasts. Moist and dry, temperate and tropical. Some of these things are aquatic. They're almost ubiquitous, the bryophyta. They're almost found everywhere. You just have to look for them. Actually, if you walk around campus and just look in the cracks between the cement, lots of times you'll see little mosses just sitting there, or even sometimes on the top of asphalt in little cracks, anywhere where they can get a little bit of soil and they can put their rhizoids in there and attach. They'll go ahead and spores will germinate and they'll grow. But you gotta look for them, they're small. 9,500 species, give or take, that's what your book says. And the genera that you'll be looking at in lab, Polytrichum, Sphagnum, which is peat moss. And this is the one where we'll do the life cycle minium, but it's basically the same as Polytrichum. So this is pronounced minium. Mini means small. So these are real small, kind of little mosses. That's the genus name. So minium is how you can say it. It's kind of like an I after the M there. What do I have up here for you to see? These are Archegonia. So they have a stalk, and they have an egg in the center. It's kind of up halfway up the structure. The whole thing is Archegonium. And then they have at the top here what are called neck canal cells. Neck canal cells help to guide the sperm down to the egg, which is sitting halfway up that little structure there. So that's called this is the archegonial head. These are Archegonia. There's an egg in there, and that's the female gamete. And then all these really long ones next to there, those are paraphyses. So those secrete mucilage, just like here. These ones here are paraphyses, just like in the brown algae. Remember we had paraphyses. They help to keep the zoospores, the sporangia, moist. It's the same thing here. These things are up on land, and when it gets hot, like today and tomorrow, and the sun is beating down upon them, they don't want their reproductive structures to desiccate, to dry out. That's what that means. So they will produce mucilage here to protect and cover these. These are antheridia that you see here. They make sperm. These are antheridial heads. So this is what these look like from up above, surface view. And they look like little diamonds. So looking at them from surface view, they're kind of cut, as you see here, they're cut more or less like little diamonds, except that they're brown. And they're brown because there's so many sperm being made in that antheridial head on that male plant. So many sperm in the antheridia and so many antheridia that it discolors it brown because the sperm are brownish, light brown in color. So you can actually visually see them by looking at them. What do the female Archegonia heads look like? They look like this. The female Archegonia heads look like this. They just look like regular mosses, like you see here. It just looks like a regular moss. But then the males, you can see that they're very different. So these Archegonia that you see here are hidden down in the leaves there, kind of protected down in there. There's two forms, two mosses. There's what they call cushiony and feathery. And they have quotes around them because those don't really mean anything. It's just there's kind of two basic forms. One is the typical moss-like thing you see here, which I just showed you a picture of. And then the other one is feathery, which means pinnate. And so it looks like a feather. Like you see here, it's kind of flat, looks like a fern almost, like you see here. So it looks a little bit feather-like. They call that feathery form. These are just close-ups of the vascular tissue. So this is if you take a cross section through a typical moss, you're gonna see in the center that there's some water conducting surrounded by carbohydrate conducting cells. So this right here is what we have over here. This is the electron microscopy, and this here is just a light microscope. But you can see these ones in the center look a little different from these ones that are smaller and have thicker walls on the outside. These are the water conducting cells. These are the carbohydrate or leptoid conducting cells. And here's what they look like when you do what's called a longitudinal section from top to bottom. You can see how slender, how narrow they look. So we have a little bit of some advanced traits here. We've got some stomata, and we've got some conducting cells. So we've got a little bit going on with this group here. There's three classes to the Bryophyta, three main classes in the Bryophyta. The bryids are the most common. So this is example would be Minium and Polytrichum. All three of these classes look a little bit differently structurally. They have a little bit different, but also genetically they've shown that these things are different. So you can see that just by looking at them. So Protonema, anybody remember what a Protonema is? Last time? Did I give you that? Looking back through your notes. Protonema are the young gametophyte plants. So in the Bryophyta, it doesn't just grow, it's almost like a metamorphosis here. It doesn't just grow straight into what we call a moss, like these pictures I just shown you. It starts off as a filament, and then it will produce a bud, and then the bud grows into what we call a moss. But this little filament is called a protonemata, plural. Protonema is singular, and they look just like green algae. So when you look at these young Bryophyta, these young mosses, and you're looking at the bryid there, you're going to see what looks like Phytogyra or Elothrix, looks like green algae, until it forms a bud. And once it forms a bud, that bud can then grow into a typical moss that I've just been showing you pictures of that. Leafy gametophytes develop from minute bud-like structures. So that's what we're just talking about. The second group is called the peat mosses. The peat mosses, or sphagnum, is the genus. They have protonemata, or protonema, with plate of cells that's one layer thick. Gametophytes have 5 little clusters of branches per node. A node is where branches or leaves come off. So there's 5 that come off per spot. And then they have a unique type of spore dispersal. So here it's showing you, this is it here, this is sphagnum, peat moss, and here it is here. They show you what happens with this thing here is one of these purple sporangia. So here's the seta, that's the stalk, that's part of the sporophyte. And then the little dark purple round thing at the top, that's the sporangium, that's the capsule. And it has a little lid on it. The lid is called an operculum. And the operculum is like a covering. It's like in a bony fish. So if you've had zoology and you studied fishes, you learned about the operculum. It's made of bone and it protects the gills of the fish. It's the same thing here. This is a little lid. It's a little protective cover. And it's protecting the spores that are on the inside here. So it's called an operculum. But it's really just a lid. So when the pressure changes inside of here, this is Boyle's law. So if you shrink the size of a bag, so you got a big bag full, if I got a trash bag, if you fill up a trash bag with air and it's this big, and then you squeeze the trash bag and make the volume of the trash bag smaller, let's say we go from this big to that big, the pressure inside there increases. If you decrease the volume of that bag, your lungs work the same way too. So if the bag is this big, the pressure is not going to be so great. But the moment we squeeze that and reduce the volume of that bag, the pressure increases. And that's what happens here with sphagnum, is that the pressure inside here increases because the size of the bag you can see here is reduced or decreases. And as the pressure builds in there, it blows off the operculum, the little lid, and the spores go flying out. And you can actually hear it. So it makes it a little explosive, tiny, baby little sound that ants can hear. Or if you want to hear it, you can lay down in the forest next to a bunch of sphagnum and sit there for a few weeks and listen to little operculums blowing off of them. But that's what happens there. So this is what it looks like when it's fresh, and there's kind of the progression of that. We have some dead, dried out sphagnum in the back. The stuff doesn't look all that attractive. It looks kind of like moss. When it dries out, it discolors. It turns kind of you can see a kind of a light tan, kind of this ugly color that I have for a shirt here. We used to have a name for this when I worked in the city of San Diego, but I won't say that. This is sphagnum. This is peat moss. It's used all over the world for different things. Peat moss is used for burning in Northern Europe. They just dig it out of the ground and they use this for fuel, for burning, for keeping their houses warm in the olden days. It's used for horse bedding, so they'll use it in horse stalls for bedding because it's kind of soft. For a while there, they started making bandages out of peat moss or sphagnum. The reason you make bandages out of that is that's much better at absorbing blood than a piece of cotton will ever be. But it turns out people didn't really like that. You go into an emergency room and you have this tan-colored, discolored moss thing being shoved onto your face or onto your body. People freak out with that. They want clean, pure, sterile-looking cotton. They're more cool with that than having peat moss shoved on their face or something like that. That stuff can absorb about 20 times its weight in liquid. And cotton can never do that. It just doesn't work as well. It's very acidic stuff where it grows, and so that acid can kill bacteria, which is also another thing. So if you ever get wounded and you're out in the wild and you don't see anything to absorb the blood because you just got bit by a bear or something, go look for some peat moss, shove it on there, and it'll suck that blood up real quick for you. That's the same stuff that they put in almost all gardening soils. They put peat moss in there. And why would you want to do that? It's the same thing as the blood. Is that if you're like me, you have plants at home that you only want to water once every 2 weeks. So peat moss, if it's in the soil, it will hold that water. And it'll hold lots of water. Like I said, about 20 times its weight. So if you put a bunch of water in there and you forget about the plant for a week or two, it's going to hold on to that water. And what are the roots of the plant going to do? The roots of the plant will then suck the water out of the peat moss. And so it kind of holds the water there and just holds on to it until the roots need it and suck it out and use it. So that means that you can have peat moss in your soil and your soil retains water longer, which means you don't have to water your plants as much when you do that. So that's what I do. Put a little more peat moss in my gardening soil. So it's interesting stuff. It's used for a whole bunch of different reasons. You'll see the sphagnum back there. Look at it. The final group is the granite mosses. They grow on granite rocks, and the name is the Andreid class. A little bit different in its anatomy. So the protonema with 2 or more rows of cells. The capsule also splits open at maturity. That's dehiscence. But it does it like this in strips of 4. So it looks like a little oval-shaped structure. When it dries out, it'll split in 4 sections. And when it splits in 4 pieces, you can see the little dots here. And it goes out, the spores fling every which direction. So it's for dispersal. You want those spores to disperse in the air. The wind will take them. Rhizoids occur in 2 rows, so all 3 of these groups are very different. And the final part, mountainous or arctic regions on rocks. You have to really go looking for these things. Climb up in the Sierras, go looking for these things if you wanted to find them. They're small, kind of leaf-like, very thin fillets. Probably have some trouble finding these things. What do you think? Extra credit? I used to send people out for extra credit for the next group that we're going to talk about, but I realized that was kind of torture for most people because they only come out in spring, and they look like grass. They're about that tall, and you'll see some in the back. They're hard to find even if you're standing on them. So we have your face right down. So I tend not want to do extra credit anymore on those things. Even if it was spring, it is torture. Okay, so the life cycle that we're going to do is this thing. I'll leave it up here for a tiny bit, and then we'll gotta get ready here, put my shirt out. This is hard work. I'm going to start it over there, but then we'll get a little this way, then I'll take the screen up. So I can use the board here. So this is, I'm going to give you minium, or polytrichum. They're kind of the same thing. They look very, they're different genera, but they have the same life cycle. Minium's the slide you're actually looking at today. That's kind of what you see here. Minium polytrichum, that's the same thing. So what it is a heterothallic. Heteromorphic. Sporic alternation of generations. So heterothallic means that the males and females are separate, which they are, different plants. Heteromorphic means that the two generations look different. The sporophyte looks different from the gametophytes. Gametophytes live a long-lived dominant plant. They live a long time. They're the green things you can see here. Here's some females you can see here. The sporophyte's a little thing that grows out of it after fertilization. It looks very different. So if we start that over here, here's some soil. Got some rhizoids in here. And the plants have their little phyllids. So these green things that you see here in the soil, these are the female gametophytes. They're haploid. So the female gametophyte is haploid. There's two of them there, I'll just say female gametophyte. What happens is we've got this thing here goes out of there, it looks like that. This is the sporophyte. Which is 2N. I'll just put the line pointing to the whole thing. So in black ink here we have the sporophyte, so we can keep track of that. Now so the sporophyte's going to grow right out of the female gametophytes. The sporophyte's got those three parts we talked about. There's the foot, which is down here. There's a little stalk, which is called the seta. And then on the top, this is the sporangium, this is the capsule. I'm going to give it a little operculum there, which I'm going to magnify for you in a second here. sometimes, well, all the time, but then it falls off. There is a little covering that protects the sporangium, or the capsule, you see up here. And you see here, this little covering is actually haploid. It's called a calyptra. So in some of these that you look at, there's a little covering over it. I'm just going to put it as if it's covering it here. This is called the calyptra. It covers the sporangium there. So that sporangium, we can magnify that. This is theta. And this whole thing is the sporangium. This little thing here is the operculum. That's the lid. And the spores are inside of this thing, this little capsule there. The other thing that's on it is there are teeth that go around the mouth. That's actually what it means. These little teeth are called, this part of the peristome, means around the mouth. So the teeth there that you see, called peristome. And when they dry out, then the operculum pops off. So the peristome changes shape as it dries out, and you see it here. These are the teeth that moist, and then when they dry out, they change shape, and then the operculum pops off, and then the spores come flying out. So that's where we're at up there with the operculum, the little lid, and the teeth are really holding the operculum in place. But once they dry out, then the spores can be released at that time. So what we have here is there's spores inside here, and once this dehisces, the spores will come out. I'm going to go ahead and lose this for a moment. So that's where meiosis takes place.